Okay, can you see my slide?
Yeah.
Can you see the pointer? Okay.
Yes.
Okay, thank you, Aza. So my name is Hongshen. I'm from Riken CBS Molecule Lab. It's such a pleasure to be here to talk about our Hippocampus paper. So the title of my talk today is Behavior Status Modulate CA2 Influence on Hippocampal Network Dynamics. Okay. So first of all, I want to give you a very brief introduction about CA2 in the perspective of physiology. So we know that CA2 is a small region which has been discovered in the 1930s. But despite its lack of mostly input from dendrogyrus and the absence of the Sorny exocrinesis, it has been typically considered a transition zone between CA1 and CA3. However, the recent advances in genetic technique have renewed our knowledge in this small region. So anatomically, it shows several unique properties. For example, the CA3 input to CA2 is relatively weak and non-plastic, and always recruit very strong free-forward inhibition, while the entorhinal input to CA2 is relatively strong and demonstrate the robust plasticity. And functionally, it shows a really active role in social memory and aggressions, and it typically shows a poor spatial coding than its neighboring CA1 and CA3, but it prefers to encode immobility and also shows strong sensitivity to the novelty. And it also has a function role to trigger the Shepard ripples. And if you block this region, it can lead to the circuit hyperexcitability. And a recent study from Hippocampal lab showing that it also has an active role to promote the social order reward learning. So all these properties not only provide the new insights compared to the traditional tri-synaptic pathway, but also making CA2 as a really elegant field to study. One of the breakthroughs in the hippocampal physiology is the finding of the place cells. So we know that if you allow the mouse to explore an open arena, and you will see a theta oscillation dominated in the hippocampal network. And if you calculate the firing rate of each cell as a function of the place, you will have a beautiful place sequence on the track. And if you, when the mouse is asleep, you will see a Shepard ripples dominant in the network, and the place cells might replay either in the forward or the reverse order, and which has been shown that really crucial for the memory consolidation. So the place cell of each region can pretty much mimic the behavior very well. However, the CA2 might display some really unique firing properties. So for example, when the hippocampal pyramidal cells, when the mouse is doing the active locomotion, most of the hippocampal pyramidal cells prefer to fire at this stage, while the CA2 pyramidal cells fire more during the periods of the immobility. On the other hand, well, most of the cells positively modulated by the Shepard ripples, but a subset of the CA2 cells are actually showing a negative modulation. These features of CA2 suggest that the CA2 might provide a unique code as a readout for behaviors. So in this work, we ask how the CA2 influenced the hippocampal network under the different behavior epochs. So to answer this question, we take advantage of the chemogenetic approach. So the control mice was injected with creatine and cherry, while the DRAT group was injected with inhibitory DRAT receptor binding with and cherry. So the mice were allowed to explore on two distinct linear track sequentially. And so the sleep one is defined as a pre-CNO session and then followed by linear track exploration termed as LT1. And after which the CNO was injected systematically, followed by a novel linear track. And finally ended with a post-CNO session. So we first examined how CA2 can impact the sharper ripples when the mouse is in sleep stage. So we calculate a correlation matrix for the two groups using the data derived from the Perry-Sharper ripples firing rate curves of different regions, including CA1, CA2, and CA3. And the left side showing the control in the pre and post sessions, and the right side showing the DRAT in the pre and post. Well, the call chart here shows the correlation within and between the regions. What you can see is like in the post CNO session in the DRAT group, the structured patterns was totally lost in all the hippocampal regions following the CA2 inhibition, accompanied with a significantly lower correlation value, both within and between groups. And this suggests that the temporal coordination activity during sharper ripples is totally disrupted in the inhibition of CA2. Besides the spiking activity, we then ask how CA2 inhibition impacted the hippocampal oscillations. So we observe a significant power reduction in both the ripple and low gamma band, which is consistent with the previous work from Dudek lab and others' work. Well, a little bit different from others' findings that showed the ripple-associated increase of slow gamma is from the overlapping of ripples. We actually observed the slow gamma emerges independent of the ripples durations in control mice. Because of the time limit, we can touch this a little bit more during discussions. But in general, we find that even under the CA2 inhibition, the ripple and the gamma amplitude still positively correlated, suggesting the temporal structure is still maintained at some level. But when we examine the coupling between the gamma and the ripple in the fine scale, we actually observe a significant reduction in both the amplitude correlation and the phase amplitude coupling. Further, we observed a positive correlation between the modulation strengths of this coupling and the replay score for the control mice. Well, such trend is totally abolished in the DRAT group. This suggests that the inhibition of CA2 not only has a function to weaken the gamma ripple coupling, but also actually abolished its associated memory replay. So after examining the CA2's functions on sleep ripples, we next sought to test its role in high velocity locomotion and the pulsing epoch. So we first quantified animals' behavior using the speed criteria when the mouse traversing on a novel linear track, which you can see here that it shows different colors here denotes different behavior epochs. So during the high velocity locomotion period, the control mice, what you can see here is it developed the place field very rapidly, while the DRAT mice display a really delayed emergence of the place field, and the stability is much poor. And for the pulsing status, we first quantified the hippocampus CA1 cells as two groups during sleep, based on its firing features, either positively or negatively modulated by ripples. For example, here it shows the P cells in control and N cells in control, and here shows the P cells for dread and N cells for dread. Then we track the P and N cell activity during the pulsing when the mouse traversing on the track. And we calculate the ratio, which is defined as the firing during pulsing divided by the high velocity epoch. Well, the control mice showed a really comparable firing between the high velocity epoch and pulsing for the N cells. Well, if you look at the dread group, it shows a really clear shift away from pulsing. And this suggests that the silencing CA2 not only has a power to slow down the place field emergence during high velocity, but also reduces the CA1 excitability when the mouse is pulsing. Finally, we use a convolutionary neural network to decode animals' location while using the unsorted spike data. So the left solid line indicate the real trajectory when the mouse is traversing, and the yellow part indicate the model predicted locations with a different color here denote the different behavior epochs. And we use a loss here to quantify decoding accuracy of each region, with a higher value here indicating actually the worst performance. In the control mice, you can see that CA1 displayed better decoding during high velocity than the pulsing. This is predictable. And it also shows a better decoding during pulsing than high velocity in CA2. This is probably due to the either ramping activity of CA2 or the N cells defined by Lauren Frank lab, which showing like these type of cells actually activated more during pulsing. While in the dread mice, interestingly, no such difference in decoding between behavior status were found in all regions, and this suggests a function loss of state dependency after the CA2 inhibition. So as a summary, in the study, we find that the dread-mediated inhibition of CA2 impact the hippocampal network dynamics in a state-dependent manner. During sharper ripples, the CA2 inhibition reduces the network synchronization and weakens the coupling between gamma and ripple, and abolish the associated memory replay in CA1. While during the active locomotion, the inhibition of CA2 slowed down the emergence of place field significantly, further reduced the lap-by-lap stability. And finally, during the pulsing, the CA2 silencing reduces excitability of CA1, especially for N cells, and further, the decoding results showed functional loss of the state dependency. Finally, I want to thank all the people from Monkey Lab who provide the crucial input, and also the GSPS grants help to help me boost my projects. Yeah, this is the end of my talk. Thank you.